The development of the nervous system begins early in embryogenesis and is one of the most complex and critical processes in human biology. It involves a tightly regulated sequence of cellular events that transform a simple sheet of ectodermal cells into the intricate network of the central and peripheral nervous systems. It all starts with the process of neurulation. During the third week of gestation, a region of the ectoderm thickens along the dorsal midline of the embryo to form the neural plate. This thickening is induced by signals from the underlying notochord and mesoderm. The neural plate folds inwards to form the neural groove, and its edges, known as neural folds, elevate and eventually fuse together to form the neural tube. This fusion begins in the cervical region and proceeds both cranially and caudally. The open ends of the neural tube, called the anterior and posterior neuropores, close around day 25 and day 27, respectively. Failure of these neuropores to close results in serious neural tube defects. Anencephaly results from failure of closure of the anterior neuropore, while Spina Bifida results from defective closure of the posterior neuropore. Once the neural tube is formed, it gives rise to the central nervous system, the brain and spinal cord. The neural crest cells, which originate from the lateral edges of the neural folds, migrate to various parts of the embryo and give rise to the peripheral nervous system and other important structures. These include dorsal root ganglia, autonomic ganglia, Schwann cells, melanocytes, adrenal medulla, and certain craniofacial structures. The neural tube then undergoes regional differentiation. The cranial end enlarges and develops three primary brain vesicles, the prosencephalon, forebrain, mesencephalon, midbrain, and rhombencephalon, hindbrain. These primary vesicles further divide into 5 secondary brain vesicles. The prosencephalon forms the telencephalon and diencephalon. The mesencephalon remains undivided. The rhombencephalon gives rise to the metencephalon and myelencephalon. The telencephalon will form the cerebral hemispheres and the lateral ventricles. The diencephalon will give rise to the thalamus, hypothalamus, epithalamus, and the third ventricle. The mesencephalon develops into the midbrain and the cerebral aqueduct. The metencephalon becomes the pons and cerebellum, while the myelencephalon forms the medulla oblongata. The fourth ventricle lies between the metencephalon and myelencephalon. While these regions are forming, the walls of the neural tube are undergoing significant histological changes. Initially composed of neuroepithelial cells, the neural tube develops 3 distinct layers: the ventricular zone, intermediate zone, and marginal zone. The ventricular zone contains proliferating neuroepithelial cells that give rise to all neurons and glial cells. These progenitor cells differentiate into neuroblasts, which migrate outward to form the intermediate zone. This becomes the gray matter of the spinal cord. The marginal zone becomes the white matter, containing axons of neurons and glial cells. Gliogenesis begins after most neurogenesis is complete. Astrocytes and oligodendrocytes are derived from neural progenitor cells. Microglia, however, are derived from mesodermal origin, specifically from the yolk sac, and migrate into the nervous system during early development. As neurons are generated and begin to migrate, they are guided by radial glial cells. These cells extend from the ventricular zone to the outer surface and act as scaffolding for migrating neuroblasts. Disruption of this process can lead to cortical malformations such as lysencephaly, where the brain surface appears smooth due to defective neuronal migration. Axons begin to grow and extend toward their targets, guided by chemical cues in the environment such as netrins, semaphorins, ephrins, and neurotrophins. Synapse formation follows, and initially, an overabundance of synaptic connections is formed. These are then refined through a process called synaptic pruning, which is influenced by activity and experience. The spinal cord also shows segmental organization early in development. Each segment gives rise to a pair of spinal nerves, which are associated with a specific dermatome and myotome. Motor neurons arise from the basal plate, which lies ventrally, while sensory neurons arise from the alar plate, which is dorsal. The sulcus limitans demarcates these two functional regions in the developing neural tube. By the end of the embryonic period, the basic layout of the central and peripheral nervous systems is established. Further development in the fetal period and after birth involves continued growth, myelination, synaptic refinement, and functional specialization. In summary, the development of the nervous system is a precise and dynamic process involving neurulation, regionalization, histogenesis, cellular differentiation, migration, axon guidance, synaptogenesis, and myelination. Disruptions at any stage can lead to congenital abnormalities or neurological disorders. Understanding these developmental processes is essential for interpreting both normal neuroanatomy and the basis of many pediatric neurological diseases.